Hi there. I'm very happy to be here this morning. Kick off this session. This is my disclosure slide. If you have questions about any of this, I'm happy to talk about current funding sources and employers after the talk. So today I'm going to talk a little bit about the science of pathogens and antimicrobial resistance in the microbiome, specifically as it relates to livestock production and food safety. Before I get into this topic in great detail, I really need to acknowledge the support and collaboration of a lot of people. This is a really complex topic, and I'm going to be presenting some complex research today, and the only reason I'm standing up here doing that is because I have a really great team of people that I work with on this topic. It takes a really diverse team with diverse skill sets across veterinary medicine, bioinformatics, computer science, math, animal sciences, epidemiology. It really runs the gamut. And so a lot of the work that I'm going to be talking about today was really made possible by the Microbial Ecology Group at Colorado State University, where a lot of the research that I'm going to be talking about today was accomplished. And so kudos to them, certainly before I begin the talk. What we focus on in my lab at University of Minnesota and what the Microbial Ecology Group at Colorado State focuses on is really trying to use scientific discovery to advance livestock production in a manner that benefits, sort of has optimal benefits for all, and that includes humans, animals, and the environment. And so in that sort of broad umbrella of what our mission is, we often tackle this question of antibiotic resistance in AMR, and specifically how livestock production practices, such as antimicrobial use, impacts AMR in these systems. And of course, this is a very front and center topic right now, with a lot of discussion going on and a lot of movement in many different areas. And what you often hear when people talk about this topic is that this phrase AMR is complex. And I completely agree with this statement. I think AMR is a very complex topic. And the reason I think it's so complex is because there are many layers of complexity. And these layers run the gamut from human behavioral complexity, so how are humans interacting with things like clinicians, and how does that impact antibiotic use and then resistance? So there's a human behavioral component. There's certainly a policy and political complexity component. So I'm going to pretend today that those things are taken care of, and I'm going to talk about two layers of complexity that we work on a day-to-day basis. And those layers of complexity are at the systems and the microbial level. And so I'm going to dive into each of these layers of complexity a little bit further and sort of talk about how these layers of complexity impact our research and how we're trying to use new tools to deal with these layers of complexity. So the first layer, which is the systems layer, I'm not going to spend a ton of time on because I think everyone in this room probably has a firsthand, day-to-day, deep appreciation for how complex the systems that we work with are. And so when I, as an epidemiologist, think about livestock production and antibiotic resistance from a systems complexity perspective, I think about all of the areas in our society where we sort of unknowingly or knowingly modify microbial populations, where we are sort of anthropogenically influencing microbial populations. And oftentimes we do this unwittingly, but the areas where we do this that I think about are food animal habitats, places like farms, feedlots, slaughterhouses, obviously our human habitats, hospitals, daycare centers, our homes. Actually, every day we're manipulating the microbial populations in our homes. And then other things we might not think about as much, like aquaculture, companion animals who live with us and obviously impact our microbiomes and our household microbiomes. And then also things like industrial production of things like biocides, pharmaceuticals, et cetera. So these are sort of the spheres in our society where we put this external pressure on microbial populations in some way. And then the question becomes, if you're looking at this from a systems level, how do these bacterial populations, and of course those populations include resistant bacteria and they include resistance genes, how do they flow between all of these different areas? And that's when it really gets complex. So I don't really want you to focus on the details of this slide, but the point is that there are all these diffuse pathways by which these microbial populations can be transmitted between the different spheres in our society. Today I'm really going to focus maybe down here a bit more, and this is sort of the food chain route of transmission from and to livestock production systems, and so I want to emphasize that these are bidirectional flows. I've sort of focused everything coming into the human habitats, but certainly these are all bidirectional pathways or multi-directional pathways. So I'm going to focus down here on the food chain more specifically, but of course as humans, we come into contact with our environment on a daily basis, and that can include things like water, soil, and air. So this is the complexity of the system, and of course, I'm sure you can appreciate that when we try to tackle this, it becomes very challenging. So next, I'm going to get into the microbial complexity of the system, and this is where I want to spend some time talking about some of the research that we've done recently, leveraging a somewhat new technology in a new way. So microbial layer of complexity, of course, is overlaid on everything. It's overlaid on all of this. There's microbes in every part of our world, right? And so that in itself is a complexity. And I want to take a step back before I talk more about the details of that complexity to sort of put us on the same page. So research methods that have been used to look at antibiotic resistance for many decades have been based on culture. And so what I mean by that is we take a sample, we isolate a certain bacteria, and that may be a pathogen that we're interested in, or that may be a so-called indicator bacterial species like non-type specific E. coli, something that's fairly ubiquitous that we can use to try to understand resistance dynamics in a given environment. But what we know is that the reality is that these microbial populations that we're trying to understand are extremely complex. So this is a pie chart on the right-hand side taken from data that was generated from a single feedlot steer, the feces of a single feedlot steer, and in that we identified over 1,000 unique bacterial species. So that's the microbiome. I think we're all aware of that it exists, but it is very complex. And so when we first dove into this topic, our question was, when we're looking at maybe non-type specific E. coli, which is this one little slice of this big ecosystem, are we really representing the dynamics of this system in an appropriate, comprehensive way? And I think we can agree that that's a question we need to answer. And so we tried to set about doing this and trying to capture the complexity and say, okay, do we get different insights when we're able to look at this entire population? Okay, so we have two layers that I've now sort of presented to you, two layers of complexity. How do we tackle them? And so this is where the approach that we take in our research group is to combine metagenomics and epidemiology. So I'm going to talk about each of these and then give some case studies of how we've used a combined metagenomic epidemiological approach to try to tackle some of this complexity and then answer some actual questions about how our livestock production practices might be changing the total resistance population in different settings. Metagenomics is really made possible by next generation sequencing machines, one of which I've pictured here. I'm going to walk you through very quickly through a traditional metagenomics workflow. So we take a sample, and that can be anything, and we extract all of the DNA from that sample. We do not enrich for anything. We don't do any culture. We don't do any PCR. We're just taking all of the DNA. We then fragment it into short pieces of DNA. That's called library preparation. And then those short pieces of DNA are then put onto a sequencing machine, and the sequencing machine just goes through those short pieces of DNA and spits out the A, G, C's, and T's sequences of those short fragments. And then those are called sequence reads. So if you hear the term reads, that refers to this short strand of DNA and its sequence. So it spits out this sequencing, this large text file that just tells you sort of all of the sequences of these short fragments of DNA in your original sample. Okay, so what does that mean? Well, some people call this big data. I work with computer scientists, both in industry and academia, who say this is definitely not big data, but this is what we generate. So we generate about 1 gigabyte worth of data per sample. So for a modestly sized data set, you generate terabytes worth of just raw sequence data. So we haven't processed it at all. So how do we process it? Well, we take those short reads, those short pieces of DNA, and we look at the sequence, and we basically just try to match them to known resistance gene sequences that are publicly available in the literature. That's called alignment, if you've heard that term, but it's basically a matching process. And every time one of those pieces of DNA matches to a known target, we say, okay, we identified resistance gene X, and we count that. So that gives us the resistome. So the total repertoire of resistance genes within a metagenomic sample and the relative abundance of those resistance genes. This is totally analogous to a microbiome analysis using metagenomic data, where we just take each of those reads and we match them to whole genome sequences of bacterial genomes, and we count. And so that's the microbiome. So we do both of these things with these data, and we do more actually, but I'm going to talk about these two things today. We also do this with pathogens, right? Okay, so what can we do with these counts that we've generated, right? Well, that's where the epidemiology comes in. We really have to understand what we're looking at, right? So we combine these two, and right now I'm going to dive into some case studies to talk about what have we learned using this combined approach. The first study that I want to go through is a study that's been out for a couple years now, actually, where we went into 4 feedlots in the U.S. And we enrolled 2 pens of cattle per feedlot, and this was a descriptive study where we really wanted to understand how does the resistome microbiome change throughout the process of livestock production. So we went into these pens of cattle when they arrived in the feed yard, and we took feces, soil, and water samples from these 8 pens of cattle. The cattle were then in the feeding period. They did receive antimicrobial drug exposures, both in feed, and then a small percentage of the cattle in the study received injectable antibiotics. When the cattle were then ready to be marketed or shipped to the slaughter plant, we went back into those pens and we sampled them again. They then were transported to slaughter, where we, at the slaughter plant, swabbed the insides of the trucks that were used to transport these cattle. They then went into a holding pen where we took, again, samples from the pen. And then they went through the slaughter and fabrication process, and at the end of that, we swabbed the conveyor belt, so product contact surfaces, in the slaughter plant, and then we took the trimmings and we took the rinsate from trimmings and used those as a sample. So we have this, like I said, sort of longitudinal snapshot of the beef production microbiome or microbial ecosystem at each of these sort of points in beef production. We took those samples, and of course there were many different types of samples, and we did this process. So I've described it to you so you sort of know what we're dealing with here. And we did this matching analysis. And so we produced 4 billion reads of data across those samples that we collected. And when we matched those reads to a resistance gene database, we found that 1.2 million of them matched to one of 319 different unique resistance genes. So that was our resistome. We did identify thousands of microbial taxa within these samples, which is not unexpected. None of this is sort of unexpected, although at the time we didn't know what we would find because this was sort of a first description. What I will say about the microbiome is that, over 90% of the data, 90% of the reads in these data sets were not classifiable. And that's not unusual, actually, for these types of samples that aren't very well represented in the literature, is that you really don't get great classification rates on your microbial species. So this is all well and good. We've characterized our microbiome resistome, but what we want to know is really how does it change, or does it change throughout the process of beef production. So I'm going to first focus on this feeding period, so the time that the cattle are in the feedlot, and look at sort of what we saw when we compared the arrival and the exit samples. This is an ordination plot, and you see a lot of these when you do microbiome work, so I'm going to walk you through one of these. So on this chart, each dot represents a single sample, and I've colored them by type, so whether they were taken at arrival, exit, or holding. And the location of the dot on the graph is relative to its resistance composition, so its total resistome. And so that's called ordination. And so the dots that are closer together have a more similar resistance composition than dots that are further apart. And so the first thing you notice is that these pink dots, the arrival samples, they are clustered separately from the exit and holding pen samples. And this is pretty stark separation for a microbiome resistome study. And so this indicates that the resistome really shifted in a dramatic way throughout the feeding period, which was interesting, but sort of high level, so the question is, what was shifting in the resistome? So this chart, again, I'll walk you through it. On the left-hand side, I have the class of resistance. On the right-hand side, there's different mechanisms that can create resistance to each of these types of antibiotics. So those are the resistance mechanisms in each class. And then the black bars show the proportion of the samples in arrival and exit that had at least one of these resistance mechanisms in it. So you'll see at arrival that at least one or two handful of samples did contain each of these resistance mechanisms, right? But when you look at exit, a lot of those have sort of disappeared from the microbial population. So we call this a decrease in the diversity of the resistome. What we see that sort of persists throughout the feeding period is tetracycline resistance, macrolide lincosamide streptogramin resistance, class A beta-lactamases, aminoglycoside, spectinomycin, and sulfonamide. Okay, so I will say that in this population, the tetracycline and the macrolide drug classes were the most widely utilized drugs, antimicrobial drugs in this population. So one theory is that the microbial population is experiencing selective pressure from these antibiotic drugs, and therefore it's promoting the persistence of these classes, whereas the other classes of resistance, the other mechanisms, aren't really giving the bacteria a selective advantage and a fitness advantage, and so they're sort of just, the population is shedding these, right? That doesn't really explain the aminoglycoside persistence because aminoglycosides aren't used in cattle production in the US. So we can't fully explain these results. It's an interesting finding. The other thing I'll point out, though, is that the microbiome was shifting dramatically during the feeding period as well. And we know that the microbiome is tightly correlated to the resistome. So can we tease apart the impacts of the shift in the microbiome from the shift in the resistome? We know things that impact the microbiome in a significant way are diet, maturation, so just animals and humans growing, getting older, and then also commingling of conspecifics, so commingling with other humans, or in this case, cattle commingling with other cattle. Those things all really put a lot of pressure on the microbiome to shift in some way. And of course, this is all happening during the feeding period, right? We're shifting the diet of these cattle, or we're heating it up. They're growing, and they're being commingled. So we think that we can't really tease apart all those potential impacts from the antimicrobial use. Using this study design, right? So that's sort of where the epidemiology comes in. So what we tried to do is to tease apart these effects is we went in and we tried to do a more controlled study of metaphylactic antimicrobial use. So in this study, we went into a feedlot, and as a group of backgrounded cattle were coming in, half of them went through the chute, we processed them, we took a fecal sample, and they received an individual injection of Draxin, which is a telathromycin, so a macrolide antibiotic. In the other group, the control group, we processed them in the chute, but we did not give them an injection of an antibiotic. So that was our control group. These cattle then went into these separate pens. They were managed identically, and 11 days later, we went back and we resampled those same cattle. So our question, of course, is on day 11, well, first of all, on day 0, do the resistome microbiomes look different? Is there some reason these cattle came in looking different, which would sort of confound our results? And our second question is, on day 11, were the microbiome risostomes different for these cattle? Well, there was actually no difference in the overall resistome composition of these pens of cattle, and again, it's just 2 pens, on day 0 or day 11. So this was kind of interesting because this is a metaphylactic treatment where the entire group, every individual in that pen is receiving a therapeutic level of antibiotic. And so we would expect, our hypothesis going in was that we would see a difference in the resistome composition at day 11, but indeed we did not. We did, however, see a huge shift in the resistome and the microbiome again over time. And so again, this is a major period of transition in the life of a feedlot steer, right? They've been transported, they're being commingled, their diet is shifting, there's a level of stress from the transport in the new environment. And so what we hypothesized from this study is that those effects, those things that as feedlot producers you may not even think of as being significant in terms of shifting the resistance profile of these animals, those are actually maybe having such a large impact on the resistome that it's overshadowing any differences we might see from a metaphylactic treatment protocol. So this is kind of interesting, and I think what this really drove home to us is that the context of these livestock production systems really matters when we're talking about antibiotic resistance, because when we're talking about antibiotic resistance, we're really talking about the microbial population, the microbial ecology. And so we have to look at these microbial populations and the resistomes in this larger context of livestock production, and what are we doing that we don't even realize right now that may be shifting these dynamics, and can we use existing tools and existing procedures in livestock production to actually maybe shift these populations in ways that are beneficial versus harmful, right? And I'm not even talking about whether or not these shifts are good or bad at this point because we don't know. But it is changing, and so it might represent an opportunity for us to come in and using these procedures that we're already doing, like commingling animals, transporting animals, feeding animals. Can we actually shift these populations in a beneficial way? So this is one of the key takeaways that I'd like to emphasize today. I'm going to circle back to this study that I started with because, of course, we have these samples from the post-slaughter component of beef production. And so we wanted to really know what happens to the resistome. We have these samples from the holding pen. The cattle then go through this process of slaughter and fabrication, and then what do we see in those samples from the conveyor belts and the trimmings? Well, when we did that matching process with the sequence reads, we actually didn't find any that matched to any known resistance gene in those 16 samples from the slaughter plant. And so at first, this was a really great finding from a food safety perspective, right? Because what we're hoping is that we're not sending resistant bacteria or resistance genes out into the food supply chain. But being aware that these data sets are very novel and new and that we don't have a lot of baseline data or context about these data sets, we wanted to really check ourselves. And so we went back and thought about the study design and thought about the sample, and we said, okay, well, what are we actually sampling? We're really sampling muscle from a steer, right? And so when we looked at the data more closely, we realized that over 99% of those sequence reads in these samples was matching to the bovine genome. So it was host DNA, right? So 99% of the data that we're producing is coming from host. Okay, so we could have a sensitivity issue. So this could be a false negative finding where the bovine DNA is so overwhelming our sequencing that we just can't detect these really rare resistance genes potentially. The other hypothesis that we thought about is that, well, these carcasses are going through a process of multiple hurdle interventions where we're doing things like blasting the carcass with high heat and steam, potentially a weak organic acid rinse, to indeed get rid of the microbes, right? So these multiple hurdle interventions are targeted at pathogens, but they're things that affect all or almost all bacteria, things like high heat, steam, and acid. And so potentially this is a real finding, right? We're really finding that these multiple hurdle interventions have decreased the microbial burdens to a degree where none of the resistance genes are getting through. So we have this sort of competing hypothesis whether this finding of lack of resistance genes is real or fake. Can we maybe start, the study was not designed to answer this question, but can we use the data we have to start to maybe tease this apart? Maybe the microbiome can help us. So we went back into the data, we did a microbiome analysis, the way I described, just matching the reads to known microbes. And indeed, it did help us sort of tease this apart. So this is a busy slide, but what I'm going to walk you through it. So on the x-axis, we have the log-fold change in abundance of these bacteria comparing post versus pre-slaughter. So anything to the right actually increased during the slaughter process and anything to the left decreased. This is a measure of statistical significance where anything higher has a smaller p-value. And then each dot here is a single genus of bacteria or virus or archaea. And so what you'll see is that the vast majority of these bubbles or these dots are to the left of the zero line. So they decreased or actually disappeared in abundance during the slaughter and fabrication process. And so that fits with our theory that we're decreasing the microbial population overall. There are, however, some bubbles over here that actually increased during the slaughter and fabrication process. And indeed, some of these actually experienced a log two-fold change in relative abundance for some reason. So we looked at what these were, and I've highlighted them, and actually a lot of them are thermophiles, like Staphylococcus, Promnesiovirus. All of these guys are thermophiles up here. We have Pyrococcus as a thermophile, and then there's some environmentally hardy organisms like the Pseudomonas as Ultramonas, and then spore formers, right? So interestingly, this kind of fits with this theory that the multiple hurdle interventions are indeed shifting the microbial ecology on these product contact surfaces and on the carcass in ways that's decreasing most things, but some kind of oddball bacteria actually thrive in some of those conditions. And they don't, all of a sudden they don't have competition, who knows what's going on, but they're able to actually thrive in some of these environments. And so we thought that this was a really interesting finding and starts to help us tease apart, what are the dynamics within the slaughter plant. And I think what really drove that home to me during this project was that we really have to understand the data. You can't just take, when you're dealing with big data and new samples and new environments using a new approach, you really have to take a step back and talk to people who understand these systems and who understand the process of producing food, and who also understand all of the algorithms behind generating these results. And everyone has to come together and talk about, okay, what are all the possibilities that could be producing this result, right? And that's why it's so great to work with a team of people who are dedicated to this topic and also really intelligent, because they can help you work through all of this, and that's why I feel very lucky to be able to work with such a team. So, don't take results at face value, especially in the era of big data. I'm now going to just quickly go into some current developments. I've alluded to the fact that we probably do have a sensitivity issue with some of the metagenomic data, particularly when we're working with samples that are 99% host DNA. So I do think the multiple hurdle interventions are changing things, but I do think we still have a sensitivity issue. So one of the things we're working on is trying to increase the sensitivity of the method while not losing the ability to look at the population as a whole, right? So that's always a balancing act. And so we've designed a system that uses these baits that can actually, we can design the baits to target anything. We've done it for the resistome, we've done it for virulence factors, we've done it for pathogens, and what these baits do is you can design them to target thousands of different things at the same time. And so they go in and they enrich their targets. And what we found is that we can increase our on-target sequencing by a 3 to 4 order of magnitude to detect rare things in the metagenome. And so this is an approach that is useful if you're interested in some of the more rare things within a sample. So this is one area where we're actively developing it. We do have a publication on it, but there's more to be done here. The other thing we found when we used this method is that we actually uncover a lot of sequence variation in the rare resistome microbiome. And so what we've done is we've actually done some fingerprinting or SNP profiling using this really deep data. So this is the TET-L gene, which is a tetracycline resistance gene. And across the sort of x-axis, you have the sequence of the gene. And each of those gray bars is one of those short reads that I talked about, right, that match to the TET-L gene. And on the top panel, it's samples from a pork production. These are fecal samples from pork and then poultry. And when there's a mismatch between the reference sequence of TET-L and our sample, we have a little red dot, right? And so you'll see in the pork that we've got these sort of very clear areas where we have these SNPs, these mutations, and in the poultry we don't have this fingerprint. And so for this specific study, we were actually able to differentiate the samples that came from pork versus poultry using this fingerprint that came from the resistome. So I'm showing you TET-L, but we can do this for every gene, right? And so this probably is ringing bells in your head about PFG, MLST, whole genome sequencing. We can actually do this at the metagenomic level. And so that becomes potentially very powerful in terms of the informative value of this type of analysis. But again, we have to tie it back to epidemiology. This is like a sort of gee whiz. We can, for these few samples, we can differentiate these two sources, but from a larger perspective, it's certainly not validated or robustly tested in any way at this point in time. We're actively developing analytical methods. I've just put four large areas here where we're working on developing better computational tools for these types of data. The databases in particular are quite challenging. And so we've done a lot of work there, and like as I said, I work with some really great computer scientists and actually physicists across industry and academia to try to develop some of these methods with the biological goal of trying to understand microbial dynamics. And so that's a really fun process. With that, I'll just bring us back to this system and sort of pose the question, you know, do we think that we can somehow merge these two layers of complexity, the microbial populations and the systems, to get a really better, a much deeper understanding of how what we're doing as humans is impacting microbial populations across this system? And I think that that's really sort of gets at the heart of some of the terms that irk me a little bit, like reservoirs and hotspots. Well, what do we really mean when we say something's a reservoir of resistance or a hotspot of resistance? I think if we can start understanding microbial dynamics at each of these points within this flow of bacteria, we can maybe start to get a better handle on, you know, where are we really, where really is the risk in this system, right? And for me, it all comes down to risk. And I haven't even talked about risk, but that's because the state that we're at right now is we're trying to understand how these results relate to risk. And so in my mind, that's sort of the holy grail of this entire exercise. And with that, I really thank you for your time and for listening. And like Kara Dow said, I can take a few sort of detailed questions, but the really hard ones we'll leave for the panel so that I don't have to answer them. So thank you.
Very nice presentation. Question for you. You're focusing on the feedlots, so that's obviously a major source for beef, and especially for primal cuts. And a lot of ground beef has come from retired dairy cows. Taking any looks, interventions there, steps in there to see what impact that might be having on resistance or other issues?
Yes, so the group at Colorado State University has an active project on cull cattle from dairy, and so that's certainly an area where we're interested in going. The trimmings were obviously meant for ground beef, but as you said, I think the cull cattle market the cull cow market's really important. So, yep, thanks for that question. Yeah.
Hi. Liz Specht, I'm from the Good Food Institute. Quick question about your controlled studies. So it was an 11-day period. Can you comment on how meaningfully that mimics kind of a real feedlot situation, both in terms of duration, as well as the fact that it was just a single injection rather than chronic exposure?
Yes, thanks for that question. Yeah, so those were actually commercial cattle, commercial pens of cattle. So they were in a feedlot, a commercial feedlot. I should have said that, I'm sorry. And we wanted to really, because in the previous study we had gotten sort of arrival and exit, so that's like a 6-month time span, give or take. We wanted to see like, well, how quick does this shift happen? So we kind of chose 11 days because what else can we had no a priori knowledge of that. And so at 11 days, we already saw the shift. So I don't know how quickly that shift happens, but it certainly happens by 11 days.
But do you think with longer time and multiple administrations, you might see those two populations start to diverge then?
Potentially, yeah. That would be the next step in that type of study, is to follow those cattle for a longer period of time. And we did another controlled study where we had a control pen and actually 3 treatment pens, and we did 26 days. And we did not see a huge difference at 26 days either. But like you said, over time, and we could certainly start to see some differences. It's a hypothesis at this point.
Sure, thanks.
Yep.
Hey, Noelle, Mark Mormon from Kellogg Company. Enjoyed your presentation. I had a quick question on the one figure that you showed, which looked like the treatment at carcass, when the carcass are going through treatment, and you had the relative preponderance of the strains of the microbiome. You had various strains, and you had the genus and species of some of them. And it looked as if the preponderance of E. coli and Pseudomonas might have gone up. And two genus that are really important, species that are very important in the antimicrobial resistance.
Yep.
Also not, thermotolerant. And I'm wondering, did you and the team look into that further to figure out why those two species tended to increase in preponderance?
Yeah, I mean, Pseudomonas, I would say, has some mechanisms to tolerate sort of harsh environments, but I take your point on that. And what I will say is that that's relative abundance within the microbiome. So the microbiome could be this large before slaughter and fabrication, and this large after. But if the relative proportion of the E. coli and the Pseudomonas is bigger in this smaller total pie, then that will be that will show up as an increase on that chart. So it's likely that the overall total counts of those bacteria are much lower, but within their microbial ecosystem, they've thrived to the point where they've been able to grow relative to the other bacteria, if that makes sense.
Okay, I think it does. So not overall numbers.
It's not absolute numbers. It's not an absolute value. It's a relative proportion, to the other bacteria in the population.
Could I then safely say that their competitive flora has been reduced, and they might have a better environment?
Yeah. So for some reason, the multiple hurdle interventions or the process that these microbes went through, most of them were almost wiped out.
Yeah.
And for some reason, those bacteria also were reduced in number, but relative to the other bacteria, they weren't reduced as much.
Yeah. Okay.
Yeah.
All right. Thank you.
Yes. Thank you. Thank you so much.